So welcome to this week's episode of Leaders on a Mission, where I'm joined by inspiring leaders driven by the impact of creating a healthy and sustainable world. Now, in today's episode, I'm joined by Brad Ringeisen, Executive Director at the Innovative Genomics Institute. Now, Brad spent his career really at the interface of breakthrough biological technologies, really for national security, and is also a pioneer in the field of live cell printing. So, with that being said, Brad, I thought you'd make a fantastic guest. So thanks a lot for sparing the time and agreeing to come on today.
I'm excited to be here. Thank you so much for the invitation.
It's a pleasure. So tell me, growing up, let's take it all the way back. What were the main kind of major influences that really helped shape the ideas and aspirations of a young Brad?
Well, most of my family is from the Midwest of the United States. So we had a very worker class and farmer upbringing. My dad's side worked in factories in Indiana. My mom's side worked in farms in northern Indiana. So it was very much hard work, get things done. Both of my parents were the first to go to college out of their families. And they both ended up getting advanced degrees. My mom got a master's in business education and my dad got a PhD in mathematics. So what that translated to was, for the first 5 or 6 years of my life, we ended up bouncing around quite a bit as my dad tried to find professorships that were stable and were a good job. So I think I moved 4 times in my first 6 years of life, which, now that I'm in an academic environment here at UC Berkeley, I start to realize how volatile and how difficult it is for early career, scientists or mathematicians to be able to make a go of it in academics. It is really challenging. And my dad, I think even way back then in the 70s, he was suffering that same problem. So ultimately, he found a home at Clemson University in South Carolina. And my mom also worked for the university. She taught as sort of a lecturer in the accounting department. And so what I'm trying to say is that I sort of bridged that world of more manual labor in factories and farms, and then bridged into this new academic world where it was not that. It was very different. And so for me, I bridged. I saw all of my cousins, all of my aunts and uncles still sort of living back in that world. And here I am in this little incubated space of academia. And granted, it was in South Carolina. And so that was unique in itself, because you have this little college town of Clemson, South Carolina. All of my friends were kids of professors. Professors would come over to our house and I'd play pool as an 8-year-old, like against professors, math professors in Clemson University. They would be betting on whether I could beat this 50-year-old math professor. Like that was the environment that I grew up in. It was all PhDs. And so to me, I think that was the biggest influence, that was what I aspired to be. Like, why wouldn't you go to graduate school? Why wouldn't you get a PhD? It just makes sense. That was what was valued in my family. My sister became a PhD in clinical psychology. I became a PhD in chemistry. And that's just what we aspired to do. And so I think in my mind, I wanted to become a professor. Because that's sort of what I knew. But as I got through graduate school, teaching is challenging. It's a challenging world. You have to try to reach so many different types of students. It was really challenging as I sort of went through graduate school. So as I got closer to graduating, I started looking at postdocs that were non-academic postdocs. And I think that was really the flip of the switch when I started seeing the opportunities to bridge into a different career path that was a non-academic career path. And that was the switch. I was like, okay, I grew up in Clemson. I got this great upbringing, being surrounded by all these really intelligent, really smart people. Intelligence was valued. It was the commodity that I grew up in. So but then I switched and went into a different kind of career path than an academic career path. And so that was really the switch in my career.
Got it. And what about in terms of finding science then, in terms of really falling in love?
I wanted to be a mathematician.
Yeah,
my dad was giving me math problems as I sat in the church pews growing up. Like here I was like doing math problems in the church pew. Like that was what I assumed I was going to be a mathematician. I went to college. I took almost all math classes. And then at some point I realized, boy, this is really theoretical. Like this is a really, like I'm struggling to find applications of what I'm really going to do for this. And that's when I flipped the switch to go to chemistry. And I saw chemistry as applied mathematics. That's what I saw chemistry as. I didn't do organic chemistry. I didn't do biochemistry. I did physical chemistry, which was basically a very mathematical version of chemistry. It was a bridge between sort of physics and chemistry and sort of theoretical chemistry. So I studied quantum mechanics. My mentor at my undergraduate was truly a theoretical. He did chemistry on paper. That was what I learned. That was what my training was in. And then when I got to graduate school, I did more experimental chemistry. And that's where I learned to become an experimentalist, essentially. But I flipped to see chemistry as an applied science. And then as I looked for postdocs, I wanted to bridge that even further. I said, look, I learned sort of some really fundamental chemistry. I now want to go into a much more applied area. So I did a postdoc essentially in biomaterials. So I essentially went and became a material scientist, which is a really much more applied field and applied area. And that kind of launched my career in a much more applied, application-driven science, which was basically the rest of my career.
Got it. Absolutely. And tell me then, like the research that you pioneered, I've written down a little bit here, the laser assisted printing, and, there were organs on chips and so on, organ printing. Give me some context in terms of the work you did in that space and why that was so important.
Well, so it's interesting because if you look fast forward to the later part of my career, I went to go into leadership at this organization called DARPA. It's the Defense Advanced Research Project Agency. It's sort of the crown jewel for the Department of Defense in terms of innovative, sort of very high risk, high reward kind of research. So ironically, that's at the later part of my career. When I went to go to the Naval Research Laboratory to be a postdoc right out of graduate school, guess what? My postdoc was supported by a DARPA project. And that DARPA project was essentially, it was called the MICE project. I think it was like micro electrical integrated circuit like program. So they were looking at ways to use laser direct write, laser printing to essentially make integrated circuits. That's what they were trying to do in the group that I joined. My mentor for that postdoc said, hey, Brad, you know what? You're a chemist. You're going to be my biology guy. You're going to be my soft material guy, not the electronics, not the circuits. We're going to try to make integrated biological circuits. And so I immediately started going right into looking at biology, looking at proteins, looking at bacteria. Ultimately, we went to mammalian cells. And this was way back in 2000, 2001. There really wasn't even a term called bioprinting at that point. What we worked on was called quote unquote direct write technologies. And they were direct writing resistive materials, silver paste for interconnects. They were using lasers to drill through vias and then sort of create these three-dimensional structures for integrated electronic circuits. My job was to try to do the corollary of that, to build heterogeneous biological systems. Could you put thin films down of extracellular matrix? Could you top that with living cells? Could you build three-dimensional structures that would mimic tissue? We were having these discussions back in 2000 and 2001, where if you now flash forward 20 years, people are still talking about those exact types of things. I think we were really probably 10 or 15 years ahead of our time and really trying to do that. And look, I pride myself. We printed viable E. coli bacteria. We printed viable mammalian cells. We did genetic testing to make sure that we didn't have any DNA strand breaks that were incurred during the printing process. And we published some of the fundamental work that people are still now showing. It still works. There are companies now that are selling that laser-based bioprinting, laser-based bioprinter, and they're using it to print skin. They're using it to print organs on a chip. And I really feel proud that I was at the early stage to be able to do some of that impactful research. But for working for the Navy, the Naval Research Lab was not the same as a Department of Energy laboratory, where you were sort of given money every year. It said, look, we need expertise in this domain. So for 20 years, people make careers at the Department of Energy laboratories in a single area that sort of is their area of expertise. At the Naval Research Lab, we were for-hire scientists. If we got money from DARPA, we would do money for DARPA. If we got money from the biothreat defense community, we would go work for biothreat defense. So we very quickly had to pivot to a lot of different areas. And I think that was a really big influence on me as a scientist as well, because here I dedicated the first 5 or 6 years of my professional career developing this amazing bioprinter, but then the money dried up. And I think, again, we were a little bit ahead of time, that there wasn't a big area of application for organs on a chip or for tissue printing at that point in time. So we actually had to pivot to bioenergy. And in the mid 2000s, a lot of people were looking at bioenergy. So we actually ended up saying that we might be able to use printing to be able to create sort of miniature fuel cells that used biology to try to generate electricity, to be able to generate energy using bacteria or mammalian cells to be able to do that. So we had to pivot from tissue engineering and bioprinting, pivot to energy. And here I am having to be the lead now to be able to go from thinking about all of these things having to do with biomaterials and what tissue matrix do you have to do, and doing all these viability assays for living mammalian cells. And we had to flip. We had to learn how to culture bacteria. We had to learn how to do electrochemical assays, set up electrochemical cells, do these electrical resistive measurements, and create completely different data sets. So we succeeded in that, and it was fantastic. And it taught me that you can't just, that you can't get too set in your ways, that you need to be able to pivot. You need to think orthogonally about different problems. And I feel like I kept that through my entire career. And look, we set power density records for how much power you could generate with bacteria. We measured how much current, how much electricity essentially can be generated by a single bacterium. How cool is that? Like, that's a pretty amazing paper that was published in Nature Communications with our collaborators at Harvard, where we measured 150 femtoamps being generated by one single bacterium. So we had a lot of fun. It was really impactful, and it taught me so much. And I'm so proud of the work that we did for the Navy at the Naval Research Lab for really 15 years.
It sounds like it suited you, that kind of flexibility and
it was a different environment. It was a different environment than a university where you form your lab, you become an expert in these labs, you think about what set of next experiments. It was very much more of a dynamic environment where we had to collaborate. We had to work across multidisciplinary areas. We had to look for funding from many different types of sources. At the end of my career at the Naval Research Lab, we were actually getting money from the Department of Treasury at the Bureau of Engraving and Printing to try to secure the next generation of banknotes. So just this huge swath of technology, and I just got exposed to a really broad base of engineering, material science, biotechnology, and I think that served me well later in my career as well.
Yeah, no, great stuff. And what about with DARPA, for instance? Was biology and Symbiote, as it relates to kind of innovation in the defense, was that something you went in to create? And tell us a little bit about the involvement of that and your job.
So I got brought into leadership into this new office that was formed back in 2014 at DARPA called the Biological Technologies Office. So prior to 2014, DARPA always did a little bit of biology. They were always interested a little bit in biosensors and biomimetic technology, and they had done a lot of neurotech where you were actually doing direct brain interfaces. So they were combining sort of engineered electrical interfaces with actual brain tissue and looking at studying brain signaling, how to control remote prosthetics and very sophisticated electronic prosthetics. They worked with people like Dean Kamen and Elon Musk and all of these amazing. So there was biology at DARPA, but it wasn't really all under one umbrella until 2014. Then I interviewed in 2016, and I think they saw that breadth of experience that I brought to the table, having thought about problems in the Department of Defense, ranging from diagnostics and pandemic prevention all the way to tissue chips. And they saw that that breadth of experience, and they said, okay, we need somebody like that in leadership at the Biological Technologies Office. And so I walked into that office, and all of a sudden I had at my fingertips anything from those brain machine interfaces to synthetic biology, to gene editing, to vaccine development, to antibodies, antibody discovery, diagnostics. Like, it was this huge spectrum of, so it really was. I know people say it all the time about drinking out of a fire hose and these types of things, but those first couple months at DARPA, I studied our portfolio of technology. And look, they made early investments in mRNA vaccines. They made early investments in synthetic biology. They had programs called ADEPT that was run by Dan Wattendorf that invested early stage in Moderna. They had a program called Living Foundries that was started by Lisa Jackson and Renee Wegersen that funded companies like Zymergen, that's a big synthetic biology company now. So they made some good bets at the early stages of that. And when I came in to be leadership, it was my job to sort of keep that momentum going and really try to accelerate, put the foot on the gas, essentially. And we did that, Simon. We really did. I mean, I'm very, very proud. I was at DARPA for about 3.5 years. We started 26 programs in my 3.5 years. We really stepped on the gas. We executed over $1 billion of funding. And we started projects in that brain-machine interface space. We started it in pandemic prevention. We started it in zoonosis, zoonotic disease, which was very clairvoyant with respect to COVID popping up in 2019 and 2020. And just, opioids, spinal cord injuries. I mean, we started some really innovative work, and I'm really proud of a lot of what we did.
Yeah, sounds like it was a great time, actually. And just when you reflect back at your career, as it were, what are the things that really stand out for you? Like one or two things that you're most, you look back on most fondly on?
Well, I think part of this is what I do now too, Simon. I'm really proud of the bioenergy work that we did at the Naval Research Laboratory. This was not my field, but I'm still really fascinated by environmental microbiology. There's just so much to be learned in sort of the microbial world. And now I get the pleasure of working with people like Jill Banfield, who is one of the world's experts in sort of soil microecology and sort of biogeochemistry. And so still trying to understand the complexity of what those systems bring to the table, the potential for biomanufacturing, the potential for climate change. And so I still go back to those early days that I was working in I'm trying to understand the basic mechanisms in some of these microbial systems in the environment and the impact that it could have for energy, for the carbon cycle, for the bioeconomy, for biomanufacturing. And so those are things that I'm really, really proud of.
Yeah.
The second thing is just generally what I built at DARPA. I inherited an office at DARPA that was sort of fits and starts. They started some programs, but they bet on some program managers, but some of those program managers end up having relatively short tenures. And it was still in an early stage at the office. The director of the agency, Steve Walker at the time, for most of my career, Steve was sort of my boss. And Steve would always say, well, BTO was an adolescent. It's out of its infancy. And Brad, you're here now with Justin Sanchez to guide it through its adolescence. And what do you know about adolescence? It's sort of a difficult period, right? It's a coming of age. It's sort of the formative years, like you're shaping the direction of what the office is really going to become. And it wasn't at all obvious at that point whether it was going to explode into and have huge impact on the world, or whether it was going to potentially collapse and the experiment was going to kind of fail. That was the environment that it was back in 2016 and 2017 at that office. And now today, you look at the office, and I think it's much closer to that explosion and the big impact than it is to collapsing. Nobody's talking about BTO collapsing. It is a solid office. It's generated tremendous amounts of strong data. You see the track record of success for generating some of the first antibodies, the neutralizing antibodies against SARS-CoV-2. You look at the investments like that were made in mRNA vaccines and DNA vaccines and how they've transitioned out. BTO's on the map. And now you see ARPA-H being approved by Congress and Secretary Barrera sort of starting to shepherd and create that ARPA-H. And they're talking about the biological office at DARPA as being a model for this new agency. So I'll put my flag in the sand there, Simon.
Right. Oh, that's wicked.
It's this office that I helped create. I think there's going to be a long legacy to that. And now if and when and as ARPA-H has stood up, I'm really proud. And I think that there could be impact that's even expanded greater in this new agency as well.
Great. Tell me about ARPA-H. Sorry, I wasn't aware of that. What is that?
Yeah, so this is something that's been talked about for a long time. I would say that actually within the first year of me being at DARPA, Jeff Ling, who was the first director of BTO back in 2014, and did a lot of the pioneering biotech and brain-machine interface work at DARPA. Jeff called me back in, I think, 2017 and said, "Hey, Brad, there's this new thing. There's going to be this," he called it HARPA, Health Advanced Research Project Agency. He's like, "There's going to be this thing called HARPA, Brad. I want your input. I want, let's talk about this. Like we're talking to the new administration. I think there's going to be something that's going to happen here." And again, there were a lot of discussions. There were some ideas generated, but it just never really got formalized. And then when the Biden administration took over, they said, "Hey, you know what? This is something that we can stake our name to. This can be something that we can say, look, we have amazing biomedical research in the United States. I think it's fair to say we have the best government-sponsored research in biomedicine in the world. Look at what has been done by all of the NIH institutes and NSF and all of these groundbreaking sort of technologies. There's no question at that. But if you look at health in America, there are some concerning trends in health in America. You have dramatic increases in neurodegenerative disease. You have dramatic increases in mental health issues, opioid addictions, obesity, diabetes. Life expectancy has kind of plateaued a little bit. So I think there are significant challenges. Obviously, cancer, we've made great strides with cancers, with immunotherapies and CAR T cell therapies, but we still have a long way to go in cancer. I know that the Biden administration is tremendously passionate about cancer. So I think the time is now to sort of try to emulate some of those types of forward thinking, orthogonal thinking, high risk, high reward types of research that DARPA is known about, and try to cookie cutter that, try to focus some of that energy to more human health than what we were able to do at DARPA. Look, we had a little bit of our budget geared towards pandemic prevention. Yeah. We had a little bit of our budget at BTO towards human physiology and sort of battlefield injuries and, those types of things. We had a little bit towards pain and trying to find alternatives to opioid, pain sites and things. But that wasn't our direct purpose. Our purpose, we had most of our money going towards operational things, bio threats. That was what our office was geared towards. What a new ARPA-H, I think, can focus on is to take the mentality, take the DARPA way of doing business and apply it significantly towards human health applications.
Yeah. Okay, great. Thank you for that. And so tell me about the new mandate then, because I think you are very much attracted by building and creating again pretty much from scratch, right? And the Innovative Genomics kind of Institute. So tell us a little bit about your kind of mandate and really what brought you to that organization.
So I joined in July of 2020. So it was right in the middle of the pandemic. And one thing about DARPA that is unique for the federal government, because look, I was an 18-and-a-half-year career, civil servant for the Department of Defense. That's what I did. But as soon as you start at DARPA, you end up, it's a temporary position. Part of what makes DARPA unique is that they want to make sure that you understand that you have a limited tenure, that you need to make an impact, that you have to run fast. And that's part of what makes DARPA special, is that you've got to start and you work and you're just really moving and going all the time. So for me, I'd been in leadership for 3.5 years. I had what I was very proud of, as I discussed earlier. And at some point, you start to think about the same problems that you'd started to think about a year or 2 years or 3 years before. And it started to feel a little bit repetitive. It started to feel like we were kind of retreading some of the things that we had thought about before. And in addition to that, I had worked for the Department of Defense 14 or 15 years prior to that as well. So I had been thinking about traumatic, brain injuries. I'd been thinking about biothreat and the emergence of infectious disease. Then COVID hits and everything gets doubled down. And we're thinking about diagnostics and vaccines. And we quick started repurposing of drugs to see if you could, find something that would work against COVID-19. And it was a really intense time. All during that, I had this opportunity. I got this email that talked about Jennifer Doudna, who was the Nobel laureate for CRISPR-Cas9 and gene editing. And there was this interesting position about to be an executive director of an organization at University of California, Berkeley. And we had funded the Innovative Genomics Institute. We had funded Jennifer Doudna in a few programs at DARPA BTO to look at gene editing, to look at protection against radiation exposures. And so I was somewhat familiar with the Institute and obviously very familiar with Jennifer's work. And I had to have a crossroads. I had to think about what the rest of my career was going to be like because I knew that it wasn't going to be at DARPA. And when I started looking at Berkeley and I started looking at the Innovative Genomics Institute, and when I met Jennifer and started to understand what she stood for, I thought, boy, this is something I could really get behind. This is something that is really exciting. And look, Jennifer is an inspirational figure. She founded the Innovative Genomics Institute to one, continue to explore and develop gene editing technologies, just the foundation of her discovery as a number one. But she also wants to look at translating that discovery into human health, into agriculture, into real world impact. That's what I started really getting excited about. And then as a third leg of sort of what the Institute's about, it's about accessibility. It's about equity. It's about how to use technology to help the world, to help those that need to be uplifted by technology. And all too often, technology leaves people behind. And you see this all the time, over and over again, where there's all this potential impact that technology could have on the lives of everyone in the world, but then you see it helping those at sort of the top of the brackets. And Jennifer, from the beginning of this institute, stated, we want to try to gain accessibility of technology. We want to improve the people's lives that are underrepresented, that need access to this technology, whether that's lowering healthcare costs, whether it's improving the accessibility of food to those that need it, to nutrition who need it, or to be able to provide crops that are resilient to climate. Because guess what? People that farm at in sort of developing countries are oftentimes at the most risk to the vulnerability of climate change. And can we make crops that are more drought resistant, that can maybe be used, that are more resistant to some of the emerging pathogens and pests that are threatening some of the world's food supply? That's some of the focus of what the IGI is doing. And so one thing led to another, Simon. And look, I mean, I'm a fan of Jennifer. And to be able to work side by side with her on a day-to-day basis, it's pretty special and it's pretty exciting. And I've learned a lot. And I think I've brought a lot to the Institute. I've brought the DARPA mentality of team building, of bringing together multiple institutions, of bringing together people from different disciplines to work together. That's what we did in DARPA. And that's what we're trying to do here by building teams to go after things like sickle cell disease, neurodegenerative disease, climate change, carbon capture, those pathogen resistant plants and crops. It doesn't just take one PI. It doesn't take just one professor's lab to do that. It takes an entire team of people to do that. And I try to help build those teams. And that's really in a nutshell what my job is.
Yeah, no, great. So how many people work at the organization?
Well, we bridge across 3 or 4 different institutions. Our home is Berkeley. We're here right on Berkeley Way, which is right adjacent to the UC Berkeley campus. But we have agreements with UC Davis and UC San Francisco, and we work with people at Lawrence Livermore National Lab, and we work with people at Gladstone Institute across the Bay. And so we're really this sort of bridge across multiple organizations, multiple institutions. And look, in the building that's behind me, which is sort of our hub, it's our home, we have about 15 or 20 PIs that are in the Institute. we don't have 100 PIs. This is a really relatively small focus group, but we do plant transformations. We do stem cell, editing. We do delivery technologies to deliver to different tissues, to deliver gene editing machinery to different tissues and cells. So that's the core of the Institute. But look, our vision, Jennifer and I's vision, is to greatly expand the impact of what the IGI does. We've actually just founded an independent nonprofit, and we are looking to expand this institute to be able to make even bigger impact. And so that's the plan, that's the vision, that's the goal. And so right now we're building coalitions of professors and of scientists that want to join that vision. We want to do additional work in carbon capture and carbon sequestration. We want to do additional work in expanding to more complex disease, to rare diseases, diseases that are neglected diseases, things like immune deficiency. We often call them N=1 diseases that big pharma is not going to go after, Simon, because there's no money, but there's still a need. Jennifer gets these really heartfelt emails from parents and loved ones talking about people that don't have any other options. So guess what? They email Jennifer and say, can you help me? And so we are trying to get to the point where we can accept clinical samples, that we can take blood samples, that we can do analysis of rare genetic diseases to see if they're editable, to see if we can help those people. So that's the future of what we're trying to build at the IGI.
Great. It sounds super exciting. It really does. Tell me though, as someone that's, you've outlined your career, earlier, the CrisprCas kind of CrisprCas9 and some of this kind of technology, that we're seeing right now, how impactful, and where is the, how far along the journey are we with this, as it were?
Yeah, it's been a pretty remarkable path. I've been around around amazing technology really for the last 5 or 6 years. I've been at the cutting edge of the best biotechnology in the world. That's what DARPA taught me. That's what the researchers that I saw at the best institutions at the world, the Wyss Institute, the Broad Institute, Gladstone Institute. These are world cutting edge leading science. What you have seen with CRISPR-Cas is a remarkably fast advancement of technology. The initial papers were published in 2012. It's been 10 years, and you've gone from an Eppendorf tube to a Petri dish to humans and to crops in 10 years. And that level of advancement and that speed to market or speed of translation, where there now have literally been tens of individuals who have CRISPR-based cures, essentially, to something like sickle cell disease, granted at a very, very high price tag right now. So we need to work on how to get that to more people to be able to lower those costs so that we can affect larger populations of people with sickle cell. But to be able to see it working in humans and for it to just be over a 10-year period of time, I can't even predict what it's going to be like 10 years from now. If I had to guess, Simon, that the pace is not going to slow down, that the billions, literally billions of dollars that are pouring into startup companies, into research labs to advance this technology, to expand to other diseases, to expand into additional crops, to be able to make impacts in things like climate and agriculture in addition. I don't think there's any slowdown that we're going to see in these technologies. It doesn't come without some pitfalls and dangers. You see some, slowdowns that have been made based on some adverse events that have occurred in human clinical trials, where you have to pause the trial and you have to do more detailed understanding of, whether these adverse events were due to the molecular reagents and the therapeutic, or whether it was not due to that, whether it was due to other genetic factors or other factors from those patients that were there. But generally, the signs are pointing towards success. Heck, there were just a release a few months ago that Intellia had been able to do systemic delivery, where you didn't have to take cells out of the body to be able to transform them and to be able to edit them, that you could actually just put the editing molecules into your body and that they could find the liver, and they were successfully able to edit the liver from that systemic in vivo delivery. So the sky's the limit in terms of what could be done with this technology. And I don't say that lightly, because I'm in the technology business, and not all technology pans out, but I really do think that the sky is pretty blue for this technology.
Great. Well, tell me, what about on the more industrial side? I completely get some of the applications, some of the areas on the human health side, but what are the thoughts towards any notable successes when it comes to agriculture or anything kind of industrial?
Yeah, well, so I think those are two separate topics. I mean, CRISPR plays a big role in agriculture. Or at least in the development, the future of agriculture. I was on stage in San Francisco a few weeks ago at the World Agritech Conference, and I was on the stage with the CTO of Corteva, and we were talking off on the side, and I think he even said this on stage. He was like, "Look, CRISPR's on the shelf right now." Because Europe still is not a very favorable regulatory environment for gene-edited crops, a lot of companies are hesitant to really go full bore on gene-edited crops as the future. United States, other countries like India are more on board, that there's a very favorable regulatory environment. And I think there is at least one CRISPR crop. I think there's a CRISPR tomato that is available now. So there is a proof of principle that agriculture can be affected positively by CRISPR. But we're still at this crossroads of whether there's going to be the full realization and the full impact. Again, from a laboratory and potential, the sky's the limit. I think you could have drought tolerance. I think you could have, wheat rust resistant, banana fusarium resistant bananas. I think you could have resistant cacao to the virus that's threatening the chocolate supply. These are things that CRISPR can do. You can increase the nutrition sort of value of a food like rice or a food like cassava. These are all things that are in the realm of possible, but we're at the crossroads of whether the regulatory environment is going to be able to allow that to happen. Okay. Yeah. And I'm on the side that I think it can be done. I think it can be done safely. I think that these edits can be done in such a way that you have positive effect on the world. So that's the agriculture world. On the sort of biomanufacturing side, these are companies like Zymergen and Ginkgo and the sort of the synthetic biology community. CRISPR is one tool in that toolkit, but there's a lot of just traditional, vector and plasmid-based technologies that you can use in those types of environments where you have a chassis, a microbial chassis that can be used essentially for industrial scale-up. And again, I think the technology there is really, there's blue sky there. You can use yeast strains, E. coli strains. You can use non-traditional chassis. You can manufacture fragrances. You can manufacture microbes that can help with nitrogen fixation. And there are companies that are now spraying these microbes on crops in place of, or to be able to reduce the amount of synthetic fertilizer that you can use. So again, I think the sky's the limit there, but these companies are also at a crossroads, because guess what? There is still traditional manufacturing, and they're in competition with those traditional manufacturing technologies, and there's different price points. And is green manufacturing gonna work out? Is it gonna compete favorably from a price point on these things? What are the markets that are gonna be impacted the most? Is it that fertilizer market? Is it the plant-based food market that's gonna benefit the most? So yeah. I think what you're seeing in the stock market right now is there's been a little bit of a correction, and that now we're at a crossroads where are these companies going to find their markets and find their products and find their niche? And are you going to see an exponential growth in these markets, or are they going to struggle? And are they going to be able to find those markets in enough time to be able to satisfy their shareholders and the desires and the timing of what their shareholders want to see? I think we're very much at a crossroads for a lot of these companies in that way as well. So it's going to be an interesting 10 years. It's going to be interesting in the human health side for CRISPR, for the agriculture side, from a regulatory standpoint, and from a technology standpoint, and from the biomanufacturing side, from sort of where the impact in those markets are going to be. So it's really a fascinating time. I'm excited to be at the nexus of it.
Excellent. No, that's great. And I've just thank you for that. Just a few kind of quick fire questions, just to kind of just to finish off with. Tell me about a leader you've really been inspired by.
A person I'm inspired by?
Yeah, a leader that you've worked with.
Leader. Well, I'm going to go back to who hired me at DARPA, Simon. Arthi Prabhatar is an amazing leader and a role model. And boy, was I inspired by her when I interviewed with her back in 2016. I think she did amazing things at DARPA. She started BTO. I think she's a visionary. She's now started in the private sector and is trying to emulate in the private sector what she did at DARPA in the public sector. And so I'm really fascinated and inspired by Arthi.
Okay, great. What about some great advice that you've received during your career that's really stood you in good stead?
Simon, that's a great question, and it's not asked very, very frequently. And so I really appreciate you asking that. And I'm going to take the time to memorialize my mentor. His name was Dr. Barry Spargo. And Barry hired me at the Naval Research Laboratory. He was a biologist, a cell biologist. He taught me everything that I know about cell biology. And as a physical chemist, trust me, that's very important. And Barry then went into leadership at the Naval Research Laboratory. So not only was he my scientific mentor, but then he became my supervisor. He gave me so much advice, but his one piece of advice that stuck with me for my entire career, when I started my own section, that was the word at the Naval Research Lab for sort of your own department, your own research group, essentially. He said, Brad, you are surrounded by very intelligent, very smart, very highly educated people. If you think you're going to just lead like you would lead in private, and where you're just telling people what to do and you're the boss and that you just are very, almost like a dictatorship, he said, that's not the way that you're going to get ahead and make friends and grow in a professional environment. He said, you must lead amongst peers. Lead amongst peers. Because as a scientist, you are always going to be embedded in peers. And I've taken that advice throughout my entire career. I try to empower those that I work with. Even if I'm in a position of leadership, I will treat everybody in that organization as a peer. Even at DARPA or now at the IGI, there are all levels of employees. There were program managers at DARPA. There was the support staff that were oftentimes PhDs that worked in the background. They never got to be the people up on stage to discuss this amazing vision that DARPA was about to launch. But guess what? Those people in the back, behind the scenes, probably did 5 times the work combined than myself or the program manager, and they got 0% of the glory. And so I would always try to go to those people. I would give them credit. I would try to pick their brains. I would involve them in the brainstorming process. And that's part of being a leader of peers. Those were my peers as much as my supervisors or my program managers at DARPA. And so I really have taken that advice. And I thank Barry Spargo for doing that. Unfortunately, Barry succumbed to a liver illness just as I was transitioning and leaving the Naval Research Laboratory. So he left us, and it really just broke my heart. But he really was one of these guys that I will never forget.
Amazing. Oh, that's quality. Final question. If you could have one wish, what would it be?
Oh, find a house in Berkeley. That is not an easy task. No, I joke. But I will pivot back to climate. I am a climate advocate. I believe that there are technologies across the board that can help fix our climate situation. I believe in technology. So my wish will be that these technologies evolve, that there is money dumped into them, that there's a willingness from governments and private industry to be able to support technologies. It can be biotechnology, it can be more mechanical solutions to air capture, carbon capture. My wish is that we get carbon capture technologies going, we get green energy going, and if we could snap our fingers in 20 years from now, we've got fusion reactors, we've got carbon, that we can control our carbon future. That's my wish for the world.
Wow. Okay, that's great. Very aspirational and very possible. But Brad, listen, what a conversation. Thank you so much for bringing so much energy and passion to the conversation and joining.
Thank you for the opportunity, Simon. It's been a lot of fun.
Yeah, great.